Hello, my name is Jason Gleghorn. I'm an associate professor in the Department of Biomedical Engineering at the University of Delaware. And today I want to tell you about some of the work that we've been undertaking as a part of the Rapid COVID Award mechanism provided by ACCEL. So as we all know, COVID-19 is pervasive in the world thus far. It's generated by the SARS-CoV-2 virus, which infects the upper respiratory tract of individuals and then progresses deeper into the lungs. Our strategy to combat this virus is to use engineering approaches to engineer microparticles that are decorated with receptors that can bind the virus and then clear it out of the upper respiratory tract through mucociliary clearance. A nice part of the strategy is it can be delivered via common aerosolized systems like nebulizers and inhalers. The microparticle system is a PEG-based polymer that can be easily decorated with different receptors. In this case, we can decorate a peptide on the surface which binds to the spike protein of the SARS-CoV-2 virus. It's also modular, so we can bind other receptors onto it to attack other kinds of viruses. And given the evolution of SARS-CoV-2 that we've seen, we can also take a multivalent approach with multiple peptide fragments, which can bind different variants of the receptor to sequester the virus. So strategy is straightforward. And to date, thus far, we focused a lot on creating these microparticles in this larger range between 5 and 10 microns. Those would preferentially deposit in the upper airways. We've done a lot of characterization and chemistry in order to both visualize it by incorporating fluorophores, as well as controlling the amount of receptors on the surface of the microparticles themselves. In addition, we also need an infection system. SARS-CoV-2 is BSL-3 required, and as we know, highly infectious in humans. And so what we've done is we've made a lentivirus from the lab using a standard lentivirus system. We've taken that lentivirus capsid and decorated with the actual spike protein that's found on the SARS-CoV-2 virus to make a pseudotyped virus that we can use for infection studies, in which case we have a virus that infects cells and transduces a red signal, a fluorescent protein in infected cells that operates through a human ACE2 receptor, spike, as well as our native lentivirus system. And therefore we can test the efficacy of the microparticles in sequestering these different viruses. Additionally, thus far, we've also been able to demonstrate that the particles are non-toxic. So whether they're no particles and the viability is similar to control particles, which you can think of as naked particles, they're just the PEG particles with no receptors on the surface, or particles with ACE2 peptides bound to the surface. Viability doesn't change, so non-toxic. We've also been able to demonstrate that we can bind virus to the particles when we mix particles and virus. And the results thus far are encouraging in terms of decreasing infection. We're wrapping up those studies. And the next steps is also to integrate in a human ACE2 mouse model so we can test infection and therapeutic potential of our microparticles in a mouse model. So we'd like to thank the ACCEL COVID program with our grant, as well as this collaboration between my lab, as well as Kathy Froman's lab at the University of Delaware. Thank you.